All right, Bears, it is Wednesday, and we are going to do our final research day to get our information together so we can put together an ad campaign to convince the government, specifically that senator that thinks small things don't matter, that we need to put some more research into the microbiomes and into fecal transplant research. So this last day, we're going to cover a lot of slides, a little bit of a mice investigation, and check out some graphs and see if we can identify the difference between the two mice. We're going to put our findings together from 2 weeks ago, and then we're going to do a little bit more research on the fecal transplant research and what is salmonella. Salmonella is something that's very dangerous, and you can get sick from it even today by eating raw chicken, and there's other ways. And so we'll do a little research on that, put together a top slide that you will take your notes, 3 to 5 notes each video, and we're just getting evidence. And then we're going to put our evidence together tomorrow for our campaign. But today is just an evidence researching. Let's get started. Once again, directions are always right here in groups. Here are your links. I will add the link to this video there in a second. Here are your videos. If you, for some reason they don't play, YouTube is a little funny sometimes, you can always log in with a personal Gmail or your mom's email, and then they will play. Let's start by reviewing our slideshow. This slide here, slide number 49, it could be 48, 40, 50, depends on how many slides you've added, if you've added a couple slides. But in the area of 49, you can see this. We have received an email. This email is coming to you, student researchers, from Mara, the head scientist. Thank you for your careful work analyzing the data of patient 23. As you know from the politician's speech, the fecal transplant procedure has something to do with feces. I've sent you a slideshow presentation to review that includes a detailed explanation of how the procedure works. I think this information will help you understand more about how fecal transplant helps cure a patient infected with harmful bacteria. We are going to, instead of looking at slides, we're going to watch a video. We also want to include your research in a press release in which we will publicly present our arguments against the senator's effort to cut our funding. In this press release, we are going to present our evidence in support of this claim. A fecal transplant can work to cure a patient infected with a very harmful bacteria, such as C. difficile or salmonella, in many different ways. We think we have strong evidence to support this claim, but we will need your help to research and write a convincing scientific argument. So what we are going to do is we're going to watch a video for our research. You can look at the slides if you'd like down here, but these, to me, are not as interesting as the video. And then we're going to come up top. We are going to look a little bit at these human and mouse gut microbiomes. Notice that they're similar. We will analyze a little bit of the data. You can come down here and notice that at the start of the experiment, 20 mice had normal gut microbiomes and 20 mice had low bacteria gut microbiomes. The biggest thing we want to take away from this is notice this is all bacteria. Look at all this empty space. Now that empty space comes from, look at this, a salmonella infection. So the salmonella infection impacts the gut biome, and we're going to get those notes from the video. So mainly we're going to do that. So let's come back to the top of our project and review yesterday's notes. Joseph did a great job. So notice, Joseph, these are notes that we can use in our campaign that we'll do tomorrow. But let's review those today. We're going to go into breakout groups in Zoom. If you're not in Zoom, that's okay. Just review your own research. You should have 4 of these done. If you are in a breakout group, there's going to be 4 people in the breakout group. Each one of you share one of the questions. Just read your question, read your quote that you've gotten. If somebody in your breakout room does not have the assignment done, then somebody that does have it done will take their place. So read them quickly and carefully. It should take less than a minute per reading, but we'll have a little introduction in the beginning. So let's do that now. I'm going to put you to the breakout rooms. Remember, each person's going to read one of these that they have. Good job, Joseph. They will read their question, and then they will answer it, and the next person will go. When you first get in there, let's have a little icebreaker by just talking about your favorite animal. Say your favorite animal and why. go around the room, and then whoever goes first, that person will go back and start here. Let's say the youngest person in the group, youngest, figure out who's youngest. You will be the manager of the group. You will keep things going. You will go first, and then you will decide who goes second. So it's simple. You go, you read yours, and you say, hey, why don't you go, Celine, or whoever you pick. Be efficient. Let's get it done. All right, let's do that now. Okay, we are back. So now we want our top slide research. Let's look at what Joseph's got going. This is what your slideshow should look like. You have directions. You have how to write a CER. He actually wrote a CER for the argument. The first argument is better because it is more informative. The first argument explains about the main topic and just proves the point about the argument. Unlike the other argument, it is shorter and less informative statement to prove a point. Second argument also states less problems about the sickness. On the other hand, the first argument states more about the sickness and its causes. It is very important to know types of sicknesses and its causes. According to my resources, my research, and my inference, I would say the first argument is more outstanding use of clauses to prove a point of causes. Excellent. Good job, buddy. You did not have to do a CER. You could have just put in 2 or 3 sentences which one of these arguments was better. But A-level people write CERs, and where does he have it? At the top. Good work, my friend. Now, there's our overview of the lesson. Let's come back up, and always our newest slide, our top slide, is the slide we're working on most recently. So let's make a new one. And here we're going to just write video notes 6 to 10 facts or evidences. So make this slide. You guys are going to make this slide. And then one video is going to be on salmonella. Now, I could show you guys something kind of fun right now if you want. Well, let's do it this way. Salmonella notes. So that's the one, salmonella. Now watch this. I'm going to take this. There's different ways to do this, but I'm just going to do it this way. So that's going to be video number 1. And then I'm going to come up here. I'm going to insert a text box. Notice I could have just clicked that. And then you just draw another text box. And then this is going to be fecal transplant notes. And remember, this is going to be evidence for your campaign tomorrow. So go ahead and let's create that slide now. Now I want to show you another little trick. So now we've got our box. If you come up here to your lines, border weight, make it 4. Now I've got a box. Do the same one for this one. And now I've got my own little box. So it's kind of fun. So that's how you can play around. Border dashes. You can do dashes if you want. Just different things you can play with to make your slide cool. You should focus on that after you have your notes. All right, let's watch our videos. Let's do salmonella.
That can infect animals and humans. The bacteria was first identified in the 1800s and named after an American scientist called Salmon. Salmonella belongs to a group of bacteria called gram-negative bacteria. They're shaped like rods and can have flagella to help them move around. There are over 2,500 different types of the bacteria. Most human disease is caused by the bacteria belonging to a subspecies called Salmonella enterica. It's estimated that tens of millions of human cases occur worldwide every year, causing over 100,000 deaths. Salmonella can live quite happily in the intestines of many animals like cows, turtles, pigs, cats, and dogs, and in birds like chickens and ducks. Other parts of the animals, like feathers or fur, or the places they live in, can be contaminated by the bacteria. Some types of salmonella, like Salmonella typhi, live only in humans. From these sources, the bacteria can spread to humans either directly or indirectly. For example, it can spread to people directly when they eat contaminated meat or animal products like eggs that are not cooked properly. Eggs, in fact, are a very common cause of salmonella outbreaks. They can also spread through direct contact with infected animals or their environments. Salmonella can also spread indirectly by contaminating water, other foods, utensils like knives and cutting boards, or the hands of someone who handles food. Salmonella is one of the most common causes of foodborne diseases in the world. People with salmonella infection can also spread the infection to others. Sometimes people can have salmonella for a long time without showing any symptoms. So what does salmonella cause? It can cause two broad types of disease depending on the type of salmonella that causes the infection. These are typhoidal and non-typhoidal salmonellosis. Let's have a closer look. Typhoid fever and paratyphoid fever is caused by either Salmonella typhi or Salmonella paratyphi A, B, or C. These two illnesses are similar. When these bacteria are ingested, they can enter the bloodstream, leading to symptoms like high fever, stomach aches, headaches, loss of appetite, or a rash. These symptoms usually occur from between 8 to 14 days after being exposed to the bacteria in typhoid fever and 1 to 10 days after being exposed for paratyphoid fever. In some cases, it could lead to serious complications like bleeding from the intestinal tract or infection of the brain. The elderly and those with weakened immune systems are at a higher risk of complications. Non-typhoidal salmonellosis is typically characterized by a gastroenteritis and is commonly caused by salmonella types like enteritidis, Newport, and typhimurium. Symptoms are usually a fever, abdominal pain, diarrhea, nausea, and sometimes vomiting. These symptoms usually occur between 6 to 72 hours after being exposed to the bacteria and can last for about 2 to 7 days. Although the disease can be relatively mild in most people, it can be quite severe in the young and elderly and those with poor immune systems. Sometimes the bacteria that cause non-typhoidal salmonella can enter the blood and infect other organs like the brain or the urinary tract and cause local complications. So, how can we prevent salmonella infection? Because salmonella is mainly a foodborne disease, one of the most important ways of preventing salmonella is ensuring the safety of food. There are a number of ways this can be achieved, including making sure food preparation areas are clean, separating raw and cooked food to prevent cross-contamination, cooking food thoroughly, keeping food at the correct temperature, and using safe water and raw materials to prepare food. To reduce the chance of being infected, high-risk foods that may contain the bacteria should be avoided. This includes raw eggs, unpasteurized milk, and raw meats. Fruits and vegetables should be washed carefully, especially if they are eaten raw. Avoid using water that has not been boiled or treated. It's very important to wash hands, especially before handling food, during food preparation, after using the toilet, and after contact with animals or those who are sick. People with salmonella infection can spread the infection as long as the bacteria are present in their feces. They should not prepare food for others if they have symptoms and seek advice from their healthcare provider on when they can go back to work, school, or childcare. This is especially important for food handlers and those who care for vulnerable people. Some people, like those with typhoid fevers, may require negative stool samples and clearance from a doctor to say they are no longer infectious. There is vaccine that can protect specifically against typhoid fever. Salmonella infections are usually diagnosed by identifying the bacteria in stool samples or blood. Other types of blood tests, like those that check for antibodies, are sometimes used but are not as good. In some instances, salmonella can be isolated from sites like brain or urine if these sites have been infected. Uncomplicated salmonella infections are usually managed by rest and adequate hydration. Antibiotics are used for some high-risk groups, complicated cases, and also to treat salmonella infections like typhoidal fevers. There are some salmonella strains that are resistant to some antibacterial medication. This is a serious public health problem because these infections are difficult to treat. For more information about salmonella infections, talk to your healthcare provider and have a look at the websites below.
All right. Okay, let's check the other one. Let's get some notes in. Remember, salmonella is a problem, and that video did a good job of highlighting that problem. It has to do with microbiotic bacteria, and a lot of people get it. There's cooking eggs. We saw the reasons. So once again, it's pretty important to study the micro world if this big of a problem is there. So you might put that in your campaign. So write 1 or 2 notes that you remember right now about this salmonella notes, about the video. 3 to 5, just quick, short notes. Let's do that now. The fecal transplant video, and then we can do some notes on that. Here we go. Well, this is an article, I forgot. So this is also an article. So here you can actually, a little easier to get your notes. You can copy and paste and or watch this video on your own. So go ahead. We're going to read this on your own. I'll let you guys get going. Watch this video. This video link is in groups. And then you can actually do the same activity we did yesterday and grab some some cited research. So you can find some things in here that you can just copy and paste. Notice it's got the same bacteria we talked about in the bacteria gut. So go ahead and use this and get 3 more notes, and I'll let you finish that on your own. And so today's due later today. Try to get it done right now. You have a little bit of time left in class. 3 to 5 notes here. If you do 3 here, 3 here, that would be 6. 5 here, 5 here, that would be 10. Go watch this video. Where's the video? It is in our groups as usual. We already watched the salmonella one, so you just have to watch this video or click this link to go find the link to that article and that video to get some notes. All right, let's get started. Have a great day.